Bem dirigir ao centro ou a câmara?
É como preferir. A câmara está ali, mas ela capta assim.
E onde é que nós temos que falar, pessoal?
Depois, quando vierem falar, podem vir aqui para esta zona para também passarem os slides. So, good morning, everyone. My name is Mafalda, and I am the team leader of the scientific department for the 8th edition of Bioengineering Week. It is with great pleasure that I present you the lecture Machine Learning in Bioinformatics, New Discoveries and Challenges, moderated by Professor Rui Henriques. Here is the QR code for this lecture, so you can scan it with your camera to earn biocoins. I'm sorry, to win one of our great prizes at the end of the week. Don't forget to submit all your questions through the corresponding section of the SBE app, so they will be answered at the end of the lecture. So I hope you enjoy, and without further ado, I hand the floor to Professor Rui. Thank you.
Thank you, Mafalda. And thank for all the team that is organizing this amazing week of bioengineering. Welcome, everyone. Good morning for those that are remotely attending this session. Would like to compliment the day, but just before introducing them, let me just open this session is Machine Learning Bioinformatics, New Discoveries and Challenges. So we have been witnessing a lot of progress in AI, as well as when we look to bioinformatics, a lot of new technologies. I would say that the possibilities for editing and or reprogramming our genome and even epigenome is becoming a reality. So how can machine learning propel these breakthroughs in healthcare, well-being, biotechnology? Okay, so I hope you are as interested and excited as I am to hear from our very special guests this morning. We have also a good special footprint. So if we start at the north of Portugal, at Braga, we meet Professor or researcher Oscar Dias from the University of Minho, then going through Aveiro and making a stop at Coimbra, Professor Joel Reis, thank you. And arriving here at home at Lisbon, we have Professor Ana Teresa, that some of you probably already know. And without further ado, I will pass the word for them to give us their stance on new discoveries and challenges in this area. Just brief introduction to Professor Ana Teresa. She's a full professor at the Department of Computer Science and Engineering here at Técnico, president of Assembleia de Escola. She was also a co-founder and CEO of a high-visibility company, Hard Genetics. I don't know, maybe you are familiar. A reference in genetic testing and knowledge acquisition, for instance, for predicting cardiovascular diseases, well-being, recommendation of personalized therapies. So without further ado, I will pass Professor Ana Teresa the word. Thank you so much for being here.
Thank you. Thank you, Rui, for the introduction. Thank you, the organization, for inviting me. Can you, where is my presentation?
Sorry, Mafalda.
Sim.
We'll stop the clock just to have the 15 minutes. Okay. So thank you all. Good morning to all of you today. So I will introduce the machine learning topic, but focusing on personalized medicine. Okay, machine learning can be used, as you will see, in many other applications. Okay. No? Okay. Okay, so then I have a clock there. It's even better. So I always introduce these talks with this image. I think that it represents a lot when we talk about personalized medicine and AI. So what can you see here? You see people, and this is a picture about health, and you don't see the image of a patient. you see the image of persons that are being advised by doctors, that are being, you are seeing a healthcare professional dealing with medical records. And you are seeing the integration of many data, biosignals, genomics, pharmacogenomics, images, clinical records. So there's a lot of data out there. But when we talk about personalized medicine, we are talking not really about disease. We are talking about health. And this is the time to invest really in the healthcare system, not in the disease care systems. That is what we are investing in all these years. So when we talk about this, and now starting introducing machine learning here. So machine learning or artificial intelligence, that is even broader than machine learning, started in many years ago, so in 1956. So we have been witnessing a number of winters and summers in this field, meaning that we see advances, then things like stay on hold for many years, and then we see some big advances after. And now we are on in one of those summers in AI. And why? Because there are three main pillars or three main events that happened that are really boosting this new field or these new advances in AI. One of the things is that we are now using new algorithms, not really based on new techniques. Most of these new deep learning algorithms that you heard about, they are based on the concept of neural networks, these techniques, they were there for many years. The point is that now we have a lot of data. And since we have a lot of data, it was possible to adjust those algorithms to be even more accurate. However, if we having new algorithms doesn't mean that we don't have new challenges. And the new challenges of these new algorithms in health, they can be concentrating in these two topics, explainability and transparency. So many AI algorithms, they give you answers, but they are not able to explain in a very clear way how they reach those results. And in healthcare, this is really mandatory. You need to explain to a patient why you are giving that answer. And this is a strong field of research, because now researchers are trying to make the techniques more explainable to be used in healthcare. The other thing is transparency. This means what? When you are building models, we need to be sure that we are using data that adapts to the population that we are dealing with. And this is really great. This is one of the, for me, the best things that AI is bringing to healthcare. Because in the hospitals nowadays, doctors are using models that are biased. Really biased. In cardiovascular is really terrible. All the models that they are using in cardiovascular diseases, they have been trained in data sets or with males, white males. And they are now being used to diagnose all the persons that arrive to the hospital. So it means that women, they are really being misdiagnosed for cardiovascular diseases. And when we start using these algorithms, they show us the bias. So we can say, wow, there is no transparency, or we are using these tools. The other thing is, okay, we now have access to big data. And everyone says, okay, big data is oil, but that is the new oil if we can use it, if we can access, if we can trust, if it is comprehensive for the field, for the topic that we are addressing. And one of the biggest problems nowadays, all over the world, and especially in Portugal, is that we have a lot of data in the hospitals that we cannot use. So data without being possible to be used means nothing. So it's a rock, not oil. The other thing is, okay, we can have great algorithms, a lot of data, but we need to run them in very sophisticated supercomputing platforms. And one of the problems that this brings is that it's not possible for all the universities or hospitals to have those platforms available at home. So we have big computational centers where we can send the data or the algorithms, but the point is, where are those infrastructures? who owns those infrastructures? Suppose that nowadays we have all the supercomputers in Russia. That would be a mess, okay? So we need to address these topics when we are talking about AI in medicine and on other fields, of course. So AI in medicine is impacting a number of areas. So image processing is perhaps the field where we can see the applications already being used in the clinical practice. So now AI tools, they need to be certified as medical devices to be used in the clinical practice. But of course, AI is being used in hospitals to optimize the processes in hospitals. I don't know if you know, but if you are going to an hospital to have a surgery at 11 a.m., all the process to prepare your surgery for 11 a.m. starts at 6 a.m. So there's a lot of things that are being prepared for you to have a surgery at 11. Then in medical diagnosis and also in medical education, of course, I'm not including here medical research in general. But in medical education in Portugal, you have very great examples in private hospitals. For example, Luz, Saúde, or CUF. I don't know if you had the opportunity to visit their training centers. They are really impressive because we can use models, simulators, robots, and not real persons. But if you go there, you think that you are seeing real persons, but they are not. They are just robots. So it's really impressive. So what happened regarding the regulation of using AI in healthcare? There are two main events that already happened 2 years ago. So in 2020, there was this white paper AI where Europe agreed that we should build AI systems, but based on our fundamental values and rights. And this means that if we are going to use an AI algorithm, we should follow those values and rights. An algorithm that was developed in China or Russia or Latin America, I don't know if they are following the same values and rights that we are used in Europe. And the other thing is what in 2021, where this Artificial Intelligence Act was presented and classified all AI applications for medicine as high risks. And this was the decision that a medical AI requires human assessment before any action. So we cannot use tools that involve AI algorithms in medicine without an assessment from a medical doctor. So now focusing on genomics, what can we do nowadays? So this picture, this represents the 22 chromosomes, and this is the result of the analysis of more than 300,000 genomes for all the possible positions in the genomes, comparing a case control study, so 200,000 persons with a high BMI and 98,000 persons with a normal BMI. And by just computing, using statistics, we can see these peaks. These peaks means what? That we have a number of positions in the genome that are contributing to this parameter, BMI. And based on this information, we can build machine learning algorithms that will learn a model to use all these data to develop something that is a polygenic risk score. And this was very relevant. Okay, this data came from GWAS studies or from full genome sequencing. And this was very relevant. And perhaps this is the most relevant picture here for the polygenic risk score field that I would like you to take home as a lesson. So this represents the frequency allele, so the frequency of a genetic variation in the genome. And this means what? That we have some polymorphisms in our genome, so some variations that are very rare, and that we have variations that are very common in the population. And those variations they can have a very low impact in our phenotype, or they can have a very high impact in our phenotype. So using the standard research methodologies that are model-based, meaning that I have a biological question, I go to the lab, I do some experiments, and I try to understand which genes are being involved in a specific condition. Based on this, on this methodology, it was possible for many years to use genomics to understand Mendelian disorders, so rare diseases. Why? Because we had access just to a small cohort. Based on that small cohort, we could go to the lab, try to understand if it is this gene or that gene that is impacting the disease. Most of the time it's just a small number of genes or just one gene. So this was interesting. So around 2007, we got to a conclusion that the genome was not explaining almost anything, just Mendelian disorders. But now, based on the genome GWAS analysis and the full genome sequences, it was possible to understand that there are many common genetic variants that have a low impact in our phenotype, but when bringing all this knowledge together, they have a very high impact. And so most of the conditions nowadays, we know that they are polygenic. Okay, why this doesn't show? Okay, so they are polygenic in their nature, and we can build, based on that information, using machine learning techniques, we can build these models where we can understand the impact of each gene or each genetic variant, the contribution of that position of the genome for a specific condition. And with this, suddenly, we could explain how the genome contributes to diabetes, to depression, to rheumatoid arthritis, to many cardiovascular disorders, but also to other things, to our weight, insulin resistance, circadian cycle. So our genome doesn't explain only our predisposition for disease. It explains who we are, our characteristics. And based on this, we can start and develop preventive medicine, focusing on healthy people and help them to avoid disease. How can we use a polygenic risk score? Nowadays, we are using polygenic risk score as a biomarker by itself. And sometimes it's very relevant because if we don't know, if we have a person without symptoms, I can use the polygenic risk score to understand the predisposition to a specific condition. I can understand many body characteristics. And for example, if I have a person with a high risk score for a high BMI, perhaps I should start following that person and advising regarding diet, exercise, to achieve that condition. Then it is useful to differentiate family members. When you go to an hospital or for a consultation, the doctor asks you about your family history. But that doesn't mean that you have exactly the same genomic background as your brother or sister. So you are different. And polygenic risk score can help on that. And then it's a very simple way to represent a lot of knowledge in just a unique parameter. So what's going on in the world regarding this? A couple of years ago, 2018, in Europe, almost all European countries signed an agreement to build a 1 million genome database to represent the genomes in Europe. Perhaps you already heard about the UK Biobank in the UK. So this is to build the initiative to build the 1 million genomes in Europe. Then there's another project that started in 2020 and finished last year that was now to start designing what this means, building the regulatory framework to understand how to produce this data, how to bring this data together. There is a big saying that is, for machine learning, we need a lot of data, but we cannot move data from healthcare data from one country to the other without the consent of a person. So if it is to bring all the genomes from Portugal to Europe, this will not be possible. And this is bringing new challenges for the AI field. Why? Because we need to do federated AI. This or distributed AI. What this means that instead of bringing the data to the algorithms, we'll take the algorithms to the data. We are building this concept of data visits. The algorithms, they go to the data, they learn a little bit about the model, they travel again to another place, they learn a little bit about the model, and they do this until they converge to a unique model. And now we are developing this project that is scaling up and sustaining. This is the GDI project. Technical is part of the GDI project. Just for you to know, the research infrastructure for genomics will be placed here at our facilities. We have the security environment where those genomes will be placed. So we are developing GDI project now, and we also started last year, October, a project in Portugal about the PTMGen, that is about the medicine, genomics medicine in Portugal. We don't have the money yet to develop this project, but now we are pushing it forward. We need to sequence 16,000 full genomes from the Portuguese population. That is our contribution to 1 million genome project in Europe. Just a short example, so I need to finish. Short example, what we are doing in this project. So suppose I have a person that is overweight. So if it is overweight, it doesn't present yet cardiovascular problems, but he is overweight. So he went to the doctor, and the polygenic risk score for coronary heart disease was calculated. And this person presented a genomic risk that was very high. So the doctor advised this person to start taking statin and also to change lifestyle to avoid triggering this risk. So, but to take statins, we need to understand if this person can take the statins. So that this is another field that it's also being really developed, and machine learning is also using, that is pharmacogenomics. So nowadays we can go to the genome and ask, can this person respond to this drug? And if it's yes, so we can choose the right statin to this patient. And like this, we can monitor the person and to avoid to trigger this risk. So this is how we will use polygenic risk scores, not only to prevent disease, but also to improve well-being. Just to finish, I'll leave you here with a couple of projects. For example, deep variant is being used by NIH in the US, was developed by Google. It's to use machine learning algorithms to better understand the genomic variations with more accuracy. From University from Toronto, Deep Genome, that is a deep learning base to also understand the association between gene variants and gene expression. Also this one, AlphaFold. I don't know if you heard about AlphaFold from DeepMind. So it's an algorithm that suddenly solved the problems of protein folding. That was a field where we have researched for more than 50 years. And now with a machine learning algorithm, we can predict the tertiary and other applications for each protein. And to finish, so we are part technical of this network, Elixir, and Elixir is the European infrastructure, so bringing together all the computational biology institutes in Europe to build this 1 million genome project and to work on genomic medicine. Thank you.
Muito obrigado. Thank you, Ana, Professor Ana Teresa, for this amazing presentation. And we will move also from the genome to the other layers and other applications. And now I invite Professor Joel Arraes. Just a brief introduction. He's a professor at the Department of Computer Science and Engineering at the University of Coimbra, has been a principal investigator of projects that also look to advances on machine learning, specifically deep learning, and can I say the discovery of new drugs?
Yes.
Yeah, and we've been also working a lot with transcriptomics and proteomics. So he will tell us more about this. Thank you.
Good morning, everyone. Very glad to be here, and I thank you so much for the possibility to be here and present my work. And of course, I want to thank the organization. They were really very professional in all the emails and all the contacts that they had with me, really, really professional. So thank you so much. Congrats. So today I'm talking about the work that I've been developing in the last couple of years, and especially on the interface between the artificial intelligence and the applications of artificial intelligence and biomedicine, especially in the use of the algorithms that are based in deep learning, mostly, for the use for the discovery of new targets, and for the discovery of development of new drugs that or at least compounds with therapeutic use. So as we have already presented, I'm currently assistant professor at University of Coimbra, and I'm a researcher in the Center of Informatics, and I'm actually responsible for teaching bioinformatics to several courses and also several different machine learning courses. And if I want to define my research, it is exactly on the use of these computer models, on the fine-tuning of these computer models for the developing, for the analysis of biological data, and of course to propose new the identification of new biomarkers and new possible drug targets. Okay, and this presentation here today, it's mostly about this. I'm not going to focus so much on the sequence data. Professor Ana Teresa already did an amazing presentation about that, but I'm actually focused in other aspects. So first of all, and also because you are students, I think in this field, and you are still in a very, very early stage of your career, I think that is very important to pass this message that I think that is currently undergoing, or actually it has already been in the last couple of years, undergoing a paradigm shift between the way that we can do research in the lab. And what I mean by this is that I think that the use of bioinformatics and computational biology and computers biology, they are changing from a data analysis paradigm where you take the data from an experiment that is already over and you do something like a post-mortem, so analysis of the data. So you have no influence in the design of the experiment itself, and you have no influence on the on the questions that you want to answer. And I think that there is this is not a new idea, but I think that is happening now. That it is that the bioinformatics and the models, they are being changed for a data-driven research, where the research starts in the computer, it starts with the algorithms, and the lab is then afterwards used to validate the hypotheses that were initially generated. And as I see it, statistics and machine learning that have been successfully used for a post-mortem analysis are now being used to design the experiments themselves. And I think there are two emerging factors that have been promoting this. And the first one, it is the availability of multi-omics data, that some of them, especially the data that does not have patient data, it is publicly available. And the other that has patient data, it can be accessible, but with a lot of other limitations, but nonetheless. This is the first factor. And the second factor is the recent developments in artificial intelligence algorithms and hardware, as Professor Ana Teresa already outlined. So I think that these two emerging factors have enabled the change in paradigm. And what I have in the rest of my presentation, it is at least one or two examples on how, based on the work that I've developed in the last years, on how I think that this is already happening. Okay, so this is the main message that I want to bring for you here today. So let's rewind. I'm quite sure that all of you already heard about ChatGPT. If you haven't, you must be living under a rock or something like that. But the main idea in here is that instead of just using machine learning to discriminate two different states, so does this image as a cat or a dog, or does this text, the person is happy or it is offensive? So instead of just using machine learning to differentiate between two different states, normal, disease, we are using artificial intelligence algorithms to generate new, never seen instances. Okay, and this is one of the best examples is ChatGPT, where we can query questions and then we can get answers that they are quite what we were expecting. And what I want to say is that this was thought to be not possible a couple a decade ago or something like that. And also there are several other tools that do the same, but for images. And of course, using the same methods and tools, that actually they are neural networks and especially transformers, encoder-decoder transformers. One tool that has a very big impact, and Professor Neutro has already talked about it, it is AlphaFold, that it is based just on the sequence of amino acids letters, so just the linear sequence of a protein. It can predict, it is the tertiary and quaternary structure of the protein. So the identification of the folding of a protein, it has been a long-term goal that it was promised to deliver a lot of Nobel Prizes, and there were some Nobel Prizes, and they didn't solve the problem itself. And I think that the major contribution over the last years, it was the AlphaFold. But I just want to outline that the AlphaFold does not explain how the folding works. It just predicts in a good way the folding for the linear sequence. And the work that I do in my lab, the work that we do, it is actually to use these generative models, so models that generate new instances that have never been seen for the generation of new compounds with therapeutic properties. And of course, what we want to do, it is to generate these new compounds biased for properties that are of interest for us. And what I'm going to talk about in this presentation, it is a couple of examples where this can be used. The first example it is quite older and it does not use this necessarily, but so it is actually for the use of RNA sequence data for the determination of HIV, for modeling the HIV immune response pathway. So in very short terms, because it's impossible for me to explain all the details in the 5 minutes that I have left, but it is something that we have the expression levels of human cells, and we are able to evaluate what are the expression levels over the first 24 hours of the progression of the HIV. And then what we want, it is to model this in a computer, in an algorithm, okay? And that was the effort that we have done. So actually what we do, it is we take the data and we use the observations from the expressions, and we try to build a model that is able to explain in a better way the explanations. Meaning, if we see this green dot that is a protein expressed in the later time phases, we want to our observations based on the transcription factors and on the proteins that were expressed beforehand to explain what we have seen. So we have a protein expressed in a later time phase. We want proteins in our earliest time phases that were expressed and can be justify this. So I'm not going to talk about the implementation details and the formulation. It's completely out of focus, but I'm more than glad to talk about that afterwards. But what we have done, it is to build a model. This is a computational representation of the model. These dots are time points, and as we can see, and as we were expecting, we were observing time cycles that they are quite well justified by the way that the virus, then the infection mechanisms work. But I think that the most interesting part, it is after building the model, and there is something that I have not said, but it is quite important. That is, this model was built with data that was already public, and it was already available, published and available. So we were not using new data from the lab to build this model. And this is just a tool that we model. But I think the most interesting part, it was that afterwards, we query the model and make the question. So which proteins do we have to block and at what time phases in order to stop the progression of the infection with the maximum efficiency. Okay, and the model returned these target proteins, and not me because I do not work specifically in the lab. But then we started collaboration after these results with the hospital, with the lab, and we bought the inhibitors for these specific proteins. And then we understood that some of these proteins could have better efficiency. They could be better targets for blocking the progression of the disease. This is experimental data in the lab. So, and we noted that some targets, they could have better results than the AZT, that is the antiretrovirus of reference for HIV. So, but I think that the message that I want to pass with this work, it is that we started from the data, and then based on the model and the results of the model, we designed the experiment, okay, and not the other way around. And I think that this was a very interesting work in the way, because it was us, the computational biologists and bioinformatics, that led the research work. And I think that this is a mindset that I think that will have higher impact in the next years. The second part of this work, it is with the use of generative models, in this case, so encoder-decoder transformers, but for the generation of compounds. And in this specific case, we were interested in generating compounds that have two different targets simultaneously. At first, it could have a good binding affinity with the adenosine A2A receptor, and it could also have the possibility to permeate the blood-brain barrier. So it's a high sensitivity to the adenosine A2 receptors in our brain. So actually, we have implemented reinforcement learning strategy where we have two different. So in this case, in the first stage, we used two different layers of long short-term memory cells. And we started by using an unbiased generator, so something that would generate, random molecules, and we train this algorithm. And then based on two different properties of interest that we had, the binding affinity and the blood-brain barrier, we were able to bias, to generate a new instance of our generator of molecules that would be biased towards these two properties. Okay, and so in the end, we started with an unbiased generator, and we went up with this more biased generator that would have better, that would be able to generate properties, molecules with properties of interest. And this was everything. All of this was made computationally. It was, I don't know if you are familiar with reinforcement learning, but the idea it is that based on the experience that the algorithms could improve themselves and learn by their own experience with the environment that in this case we simulate. Okay, so this is just some metrics, and it is full of numbers. But if I have more minutes, I would prove that everything was good with the results. And of course, it is very outlined in the paper. But I think that you can take my word, we were very happy with these results, and especially with the possibility to be able to start from an unbiased generator and to be able to show that we are able to bias towards a specific property of interest. So this was what we were able to do in the first stage. And this is some of the molecules that our model generated. And this is a very old version, almost with 1.5 years of our work, okay? Now they are much, much better. And so the second example that I have, it is exactly the same, but instead of using LSTMs, we are using encoder-decoder transformers, the same as AlphaFold and so. And we are, so the idea in here, it is to use the response of the genetics expression of our cells to guide the design of new compounds. Okay, so, and we apply this to the ubiquitin-specific protease 7, the USP7. That is a very important target in cancer, or at least not a very important, but a promising one, a promising targeting. So actually what we have, it is, once again, we took all the data from TCGA, that is data publicly available for gene expression transcriptomic data, and we train a variational autoencoder and that instead of learning a discrimination learns the distribution of the data and then is able to generate new instance. And then in short terms, it is able to replicate the behavior of our cells to a new compound. And then we evaluate the new generated compounds and we are able to obtain conditional gene expression profiles. So we are testing the response of our cells to new compounds that we are developing at the same time. So we are fitting together these two stages, okay? What we think that, or at least what we have shown that we are able to do, it is to reduce the side effects of the drug, because we are evaluating them at the same time that we are designing the drug. And so this was a very, I think that this was a very outcome from our lab. And I think that we are, I'm quite sure that in the next years we will have even more impactful molecules. And in this case, we have already generated a couple of molecules, and some of them actually they are new, but they are similar to already some already known anti-cancer drugs, and I think that we can even improve this. And yeah, so this was the main message. I know that I'm already surpassing the time that I have. So the main idea that I want to pass to you, it is that first the paradigm shift that we are using, moving from the data analysis to the hypothesis generation and the use of computational computers to do that. And the second, it is that maybe we can simulate, or the new advancements in artificial intelligence would enable us to evaluate all of these possibilities in silico before going to the lab. And I think this is actually what we are building in my lab at the moment in Coimbra. Of course, everything that I have taught and a lot of more other things, they are not just being developed by me, but by all the team that is working in these topics. And I think that they are really, really motivated and really good. So I'm really glad to have all of them in my team. So I think that I'm already over time, but thank you so much for keeping me.
Many thanks, João. So now let's go to our next presentation. Afterwards, we want to hear from you, and I will ask our very special panelists to provide recommendations and suggestions if you, in your life ahead, you want to become a data scientist with a background in biology to be part of these ongoing breakthroughs. So Joel is a professor at Oscar, at the Department of Biological Engineering at the Universidade de Minho. You're the CEO of OmniUI. OmniUI. And you work with metabolomics, microorganisms, and plants. So we will hear more about it from Oscar. Thank you.
Okay, first of all, I would like to thank you for the invitation to be here. It's always nice to know that people appreciate what we are doing and invite us to come here to present our work. The two previous guests already raised the bar really high, so we have to lower your expectations. I'm not going to talk about personalized medicine or nothing much related with medicine, just to be different. Okay, I have a little bit to talk about medicine, but not that much. I am a researcher at the University of Minho, so like most, at least some of you, my initial background, my degree wasn't in informatics, it was in biological engineering. Then I moved to informatics in a master's, and then on my PhD, I went for systems biology, computational biology, bioinformatics, and all of that. And now I teach bioinformatics and systems biology, and also have our company, which has a really hard name to say. It's for people from the outside, from abroad. But I'll tell you more about that later. So, like Ana Teresa and Joel were saying, so we have a paradigm shift. We have lots of, everyone is talking about machine learning right now in bioinformatics. And not only because we have the infrastructures, the supercomputers, and the algorithms, but also because we can see that just in the last 20 years, we have a real high price drop, and the availability of data, of omics data in this case, of DNA, is really high. So besides besides we have the ability to move on, we really have to move on from the traditional algorithms that analyze data post-mortem, like Joel was saying, and we have to make some large volumes. It's a trend. So since 2010, the publications that cite AI have more than doubled, and if we go even more further back, it's even worse. So it's even higher. The number, it's about 18 to 20 times. 50% of all the documents published that mention AI in 2021 were papers. So there was a shift. As you can see, there's a steep increase. In 2014, you can't really see here, they weren't that much, but now they raised a lot. They also, AI was mentioned, not that much, but now we have lots of papers of manuscripts that are mentioning AI, which is a really game changer. And from this machine learning since 2017, now we can see really well, it's the term that has been increasing more. So in AI, we have lots of things. Machine learning is the one that is the most trending right now, besides pattern recognition, and other terms more associated with deep learning, like computer vision, data mining, have smaller increases. And this shows us that, yeah, AI, machine learning, all of that is really trending in all areas, and science is advancing really quick with this. Like Joel said, ChatGPT, everyone has used it here, and it's a tool that most people don't understand, but that at least they know how to use it, and they are impressed with it. And I have a couple of teasers here because Ana Teresa and Joel were talking about AlphaFold, which was a really breakthrough in the last few years. But last week, so when I was preparing this, I was doing some research, and last week there was a paper from Meta, the company from Facebook, Instagram, and all of that, where they have their own version of a protein folding algorithm, which they claim that can go to the atomic protein level. To the atomic level for the protein structure. So instead of just having the three-dimensional representation of the amino acids, they claim that they can go the whole way to the amino acid, to the atom level composition. Of course, this was really published in Science, so this was last week. So this was reviewed. So this is another major breakthrough that will probably give us some more advances in science in the next years. Also, and a full disclaimer, I haven't read this, I haven't had the time yet. regarding the area where we usually work with at the University of Minho in our bioinformatics group in systems biology, specifically, we develop tools for constraint-based modeling, for mechanistic models of the organism. So genome scale metabolic models are models that represent the metabolism of microorganisms, of plants, of humans even, and they are basically networks of reactions that represent a phenotype of the organism. So we understand the behavior of the organism under certain phenotypical, genetic, and environmental conditions. So we can understand, so for instance, we can take yeast, okay, let's, based on this model, on this mechanistic model, if I try to grow him, the yeast in anaerobic conditions with this amount of glucose, what's going to happen? Is it going to produce or not? Of course it will. So we have these types of models, and usually this is the workflow that we follow. We perform an initial genome annotation. We assemble a draft of the metabolic network with several biological databases. We assemble all of these in matrices that represent the stoichiometry of the reactions associated with the genome. And then we can validate this model and go to the lab and repeat the cycle if we need to. And again, last week, there was a paper on deep learning where they automated all of this process to build all of these this type of networks. There are lots of tools that do this some automatically or semi-automatically, and now we have, we might have, because this is even a preprint, so it's not peer-reviewed yet. So this is a preprint from a journal at Springer, where they are proposing all this architecture with two tools, nice game and close gaps, where they are going to generate all of these automatically. So again, another major advance, another major breakthrough on this area. So indeed, machine learning is already revolutionizing bioinformatics at all levels, and we are trying to keep up. And I'm going to just show some of the things that we are doing in this time specifically with plants. So this is Marta Sampaio. She's exploring the synergies that we can have between metabolic models and machine learning. So one of the things that she's doing is implementing this type of multi-view learning from multi-omics data, where she's testing different approaches. The first one is basically taking different types of omics data, transcriptomics and metabolomics, for instance, and putting all them together in a matrix and creating machine learnings from this. Also taking that data and doing an intermediate step where she converts the omics data into graphs or kernel matrices, and then she trains metabolics models, machine learning models with this. And also a final, a third option of taking each of the omics independently, creating the individual ML models from this, and then integrating all of this in the end. And all of this allows us to interpret better the data, to extract underlying knowledge that wouldn't be, that a researcher looking with the traditional tools wouldn't probably get some aspects that are very interesting from this data, that might be interesting. And since we are exploring the synergies between modeling and machine learning, we want to integrate all of this. We are trying also different approaches, just by taking our metabolic networks, performing simulations, and our simulations, basically what they give us are fluxomics data, so the fluxes that go through these reactions, and then just training machine learning models to see if we can get more from that. But this is just the simplest approach. Then we have the multi-omics analysis where we can, for instance, for plants or for humans, we can take omics data like transcriptomics and create models specific for a tissue. So for plants, we can take that model, and with transcriptomics data from the leaf, we can create a model specific for the leaf, like the same way that we can take transcriptomics data from the liver, and with a model from a human, we can make a specific model for the liver. And then taking the fluxomics data from these models, from these condition-specific models, and integrating them with the other data, metabolomics, or transcriptomics, whatever, we can also create ML models that help explain better what is happening. Or in the end, just going the other way around, but taking the ML models that we trained with omics data and creating context-specific models with the fluxomics data. Also, we have João Capella, that he's starting his PhD. He started some months ago, 6 months ago, and he's going to use AI to expose the plant's specialized metabolism. So there are several questions that we can ask when studying plants, and some of the compounds, as you probably know, there are lots of compounds that come from plants that have pharma properties, and these are other properties, of course. And these are some cases of some of the questions that we can ask on the origin of metabolites that we have. So sometimes we have some pathways that we already know what's happening, but with some key steps, we don't know which enzyme should be associated with that type of reaction. In other cases, we know that these might go from here to there, but we don't know the reaction, if it is more than one even. And not, of course, if we don't know the reaction, we also don't know if there's any enzyme involved. And sometimes there are compounds that are being produced, some metabolites that have specific properties, and no one knows how they got there because there's no information on the pathways that create them. And here are the highlights, and what we are going to do is to use artificial intelligence to try to help us highlight this problem, to solve these problems. So the first thing that we are going to do is try to predict EC numbers. So EC numbers are, for the ones that don't know, enzyme commission numbers. So they're like a code that represent the enzyme activity. So not really a reaction, but it might be more than a reaction, but which type of reactions that enzyme might catalyze. And using AI, we are going to go through several steps. First, to predict if it's an enzyme or not. Then to predict the family of the enzyme. So these problems are somewhat solved. There are some models that already do this. But then we want to go further on and predict the substrate associated, the substrate or substrates that a given sequence can catalyze, can transform, can catalyze a reaction into another compound. Then we are going to use some interaction deep learning models. So besides performing this characterization, we want really to identify the enzyme substrate and the enzyme transformation interaction. So we can take a sequence and a compound, and using these models, we want to know if they interact, if there are any kinds of, even if weak interactions, if we can predict the interactions between the enzymes and the substrates, binding affinities, and all of that to try to identify some potential enzymes to fill in those gaps. And also for the last problem, we want to try to find the whole pathways that can lead to a compound using different techniques. Since for the traditional, starting with the traditional pathway enumeration techniques, when we can see all the routes that can take us from point A to point B, but also some retrobiosynthesis techniques that apply transformers where we can use generative models like Joel was mentioning before, that can create a reaction or a series of reactions that can take us from to a substrate. And besides plants, we are also working with another type of organism, which are bacteriophages. For the ones that don't know, bacteriophages, usually called phages, are viruses that attack bacteria, hopefully only bacteria. So we are talking about Midjourney, ChatGPT, and this is the representation of DALI. So it's OpenAI's image generator of a phage attacking bacteria. So you can see that there are still some years from some years from AI taking over the whole world. But regarding bacteriophages, there are lots of AI research on this. So different tools that perform different tasks like life cycle classification, host prediction, genome binning, etc. Lots of AI tools that already are studying this. And we also have a couple of these, like the ones that are highlighted there in bold. So we have an ML tool, PHDPO, which classifies eventually a given protein in a phage like a depolymerase. These depolymerases are very important because they are the ones that allow the phage to infect the host. And also the phage promoter, so basically locate the promoters in phage genomes and to know where the annotation starts. And besides this, we are with these tools and others, we are involved also in a project that is funded by La Caixa, which is a really challenging project where we are supposed to study to basically create cocktails of phages that might not exist to attack bacteria associated with resistant diseases. So basically, this is what we are proposed to do in this project. We are the AI partners, so our other partners are going to take the samples from people that are infected with Klebsiella or other bacteria that have a resistance to antibiotics. Then we, using AI, we are going to try to find the best phages to attack those bacteria based on the capsule that they have. Then we are going to basically to identify the phages that can attack the Klebsiella and mount. So using different parts of the virus, create a new phage that can be administered to the patient in the hospital to fight those infections. So this is very exploratory, of course, but it's a really interesting project and it's really challenging because there's not as much data as we hoped for and as we expected. So basically, we'll do the in silico prediction of the binding proteins, and then starting with the viral chassis, our partners at the lab will create the customized phage to administer to the patient. And now I'm okay. Of course, I'm over time, but I'm going to take some minutes also to do some shameless promotion of our company. So almost 2 years ago, we started a company, Omnium AI. Our company is a spin-off from Omnium. We work by providing solutions in artificial intelligence and biomedical data sciences. So we started in 2021, so full pandemics. Everyone was working, trying from home, and so were we. And we decided to try to go on with this. So here we can see that all of us work in silico, so we don't have a wet lab part. Basically, we are building a platform that is starting with the creation of an integrated knowledge base. We are taking information from all publicly available databases and also mining data from articles, from patents, from clinical trials, and integrating all of this in a knowledge base, which is going to be used by our platform, our AutoML platform. So this platform, Omnia, is a platform that does most of the steps of AI automatically. So the hyperparameter optimization, the model selection, all of that is done automatically based on the data that we want to train the models with. And then we want to take some AI-based optimization approaches, like we're using evolutionary algorithms or Bayesian methods to perform the model optimization. So behind Omnia, we have the model training with machine learning and deep learning models. We have different representations of descriptors and all of that. And basically, we want to apply this platform, which is a green platform that we are developing, considering all the implications of the green AI that are going on now, of having efficient algorithms, efficient computational structures, and apply all of this to some of the things that Ana-Teresa and Joel already mentioned, like designing of new molecules, personalized medicine. We have a case study that we have already published some data on developing, don't exist in nature, but are being synthesized in the Shiki Factor project that have that property of being sweet. This is, of course, just a proof of concept. So we already have some open source tools, libraries, and applications. And that's it from my part. Thank you.
Obrigado, Oscar. So now I want to turn this session a little bit more interactive. We don't have like that much time left. Just be aware that this was just a small piece of many ongoing breakthroughs of machine learning, applications of machine learning in bioinformatics. You can, for instance, if you think of bioreactors to decompose material or produce new compounds, how do we optimize them based on experience, okay? I'm talking about how to optimize those bioreactors, like the conditions, the microorganisms in there. Or if you want to go to stem cell therapies, for instance, how we differentiate is also is to optimize bioreactors that are able to produce, to differentiate skin cells in cells that are particularly hard to differentiate. Or if we want to go, for instance, to neurobiology, how do we understand the regulatory activity in our brain if biopsies are very intrusive? So can we go beyond, for instance, animal models and in vitro studies, and for instance, look at post-mortem cohorts in order to understand what's going on in our brain, based, for instance, on the regulatory activity on peripheral tissues, okay? So there are many, Joel and Oscar also touched, and as well, talked about this new therapies. Can we go even further and instead of only looking to the protein structure, can we also look to the modifications that proteins may have in terms of maturation, some molecular compounds that are added on top of the structure of the protein, so the protein can sometimes provide a more efficient function, but depending on how those molecular compounds, such as glycans, are added in those domains. If if there's something in a domain that should not be, for instance, glycosylated or something like that, maybe the protein loses its function. Okay, so this is also a project that we have with IPO at Porto, where we are looking to mark those proteins that have abnormal patterns of glycosylation, and then we can find them at the surface of cells, mark them to develop new immunotherapies. Okay, so there's plenty of things. I believe you now will hear about on the next section about neuromodulation, so stimulation therapies and neurofeedback. Okay, that's also like an area where AI is propelling a lot of breakthroughs. So I just wanted to make this parenthesis, and now I will open the session also for you to ask our very special guests your questions, if you have them. We have some questions from people that are joining us remotely, but I will first ask if anyone
They are from the room.
Ah, they are from the room. Oh, perfect. So, how can machine learning revolutionize process engineering, design and control, or substitute analytical methods? Okay, how can machine learning revolutionize process engineering, design and control, or even substitute analytical methods? Okay, so I don't know if anyone wants to start with a take on this.
I can say that was the last, so give me the first. Regarding the analytical methods, in my opinion, I think it can complement them, not just replace them. At least some of them. Having the history of analytical data that we have, it depends also on the analytical data. And taking in new information, we can can be easy to interpret results by getting the data and just comparing them, just running them to an ML model and see what were people that base we had before. Process engineering. That's again, it depends on the area because process engineering is also very broad, but that can be interesting. There are a lot of automation already in process engineering, and I think that ML can these models can substitute some of the rigid rules that those automations have, and adapt and make an adaptive control that is more in the runtime than just having a set of rules that are triggered by a given aspect.
Okay, so the question was from André. Obrigado, André.
Yes, I think we can add also that since we can use reinforcement learning techniques, so the models can really adapt in the direction that Oscar was saying, that is, they can observe the behavior and adapt in real time and learn from that. That is something that the analytical models cannot.
André, have another? No? No, I'm going to skip to the next question. So I don't know, Joel, if you want to add anything. So the next question is from Inês Pacheco. Obrigado. When you have a certain data set, how do you know which model to use? And welcome to the machine learning course. And that will lead to the highest accuracy. Okay, so which best model? You just test different algorithms, or you choose one, stick with it, and try to improve it. If so, how do you know? Do you really know that that is the best option? How do you know that is the best option? Yeah, so I don't know if you anyone wants to start.
That you can answer yourself. That is machine learning.
Yeah, so knowing the data is, so for those of you who want to really become a data scientist or have some competences at that, knowing your data is very important in order to make preliminary choices, okay? So because there's so much algorithms available and things coming out. Having said this, we may have some clues that, okay, this might work better than the other, but at the end of the day, we should be able to at least have a broad set of experiments in order to see which algorithm algorithms may have different types of performance, but may be better at improving sensitivity, other precision. So it is important to have this comprehensive view. So early in the day, it was more or less easy, based on the type of data we are analyzing, to anticipate what would be like the best type of model, or under an hypothesis what it would be. But nowadays, with the advent of deep learning, sometimes it's a little harder. So we cannot skip hyperparameterization, the optimization. It's not really a hot science. It's more like an experimentation in terms of loss functions, etc. So we are unfortunately unable to skip like this model selection and hyperparameterization part these days. But of course, it's still very important, the key to know by heart our data in order to make some decisions, because it's impossible to test everything. I don't know.
Well, or everything that we said is perfect. Or you can try to pick a model that is not very well known, but somehow trendy, and you try to force the data to the model to fit the data, and you try to get the best of it. Of course, that if you follow this strategy, in the end, you have nothing. You have no understanding of the data, and you have no understanding of the model. Even if you have the results are good, it doesn't matter. So actually, as Rui was saying, that the most important thing is to try to understand the data and try to see what are the relations in the data, what are we trying to model, okay? And only after that, you try to think if it is important or necessary or useful to apply any kind of machine learning. So if what you are trying to model is way too simple, you don't need machine learning. You build your own rules with code. You can even just implement some conditionals, ifs, and you can, you don't need anything. You don't need machine learning. If the problem it's way too hard, and if the relations in the data, they are not explicit, maybe the data is not the best one to use machine learning, because the problem is so complex. In the end, it will not be able to model that. For everything in between being way too easy and way too complex, I think that machine learning has proved to be very, very useful. Okay, but the selection of the model, it's many times influenced by what you think that is the behavior or the distribution of the data. Okay, and you can only have that after you being quite aware of what are the properties or specificities of every specific model and the specificities of the data. Okay, and then you can choose from something that is really easy, simple, like a decision tree or nearest neighbors, to something that is very complex, like convolutional neural networks or even transformers. So it's not easy and it is not straightforward, but most probably if your data is in this in-between zone, maybe even if you choose a model by chance, you'll get some results. Not the valuable ones, not the best ones, but some results. So I don't know if I...
Yeah, and validation is very important then to assess the ability to generalize and other properties. So for those of you who want to know more, we have different courses on machine learning and data science to get deeper on this. So Mafalda asked, data availability is very important in this area to establish models. Do you think that the regulations regarding data sharing are setting back new developments in machine learning? So this is actually one of projects that Ana Teresa
Not
is...
setting back, but bringing awareness that we need to be careful.
at the end of the day, what we are doing is we have the regulatory frameworks in place. A number of them are changing, especially those regarding the genomics. They were very strict at the beginning. Now they go to a model where the patient or the person can give their own consent. And let me know, everyone gives the consent. It is very interesting, because if we don't share the data, we cannot improve medicine. So we are blocking. Sometimes I try to explain that we have vaccines, we have medicines, we have many things nowadays, because there were people that went to clinical trials and shared their data. If this was not possible, we could not improve medicine. So we need to share. Of course, we need to build the regulatory going. And of course, when the GDPR was published, it was something, well, no one will share the data. It's not true. We are just improving the frameworks to allow people to be safe when they share the data. But people are sharing the data. I have a very interesting result. When I was in the company, we have this strict regulation to give the result, a genetic result, before GDPR. So before GDPR, we need to give the result to a medical doctor, and the doctor only gives the result in a consultation. With the GDPR says that the data belongs to the patient. So if the patient asks for the data, the lab or the hospital needs to give the data to the patient directly. So every day we were breaking the rules, or we are against the GDPR or against the framework, the legal framework in Portugal. We hold in a very controlled place with the doctor and all of this, and then the patients, they paste the results in Facebook. They already shared the data, so at the end of the day, okay.
Any other take? Let's move. We still have a few. If you want to ask questions alive, also be welcome. We have one, I think this one may go first as well. Is related with, Oscar, multi-resistance to drugs in cancer cells is one of the biggest setbacks in chemotherapy. How impactful will AI algorithms predict drug-protein interactions? How will AI that predict drug-protein interactions? So this is also something that I would I'll also ask, because most of your previous studies, they are.
Can you read the first part?
Yeah, sorry.
Because
multi-resistance to drugs in cancers, in cancer cells, is one of the biggest setbacks in chemotherapy. Okay, so how impactful will AI algorithms that predict drug-protein interaction be in this issue?
Well, actually, I don't know. I hope that's big, that's at least some. But I think that following my previous answer and my previous comments, I think that the idea in here is that we are able to model as much as possible from the previous observations that we have, and to be able to model this in a computer and machine learning and deep learning and the methods, the models that we use, they are very interesting for that, because they are able to model the distributions and behaviors on the data that they are not easy to identify by just analyzing the data. And so I think that they will have a big impact, but I think that these contributions, they are still on development. Okay. I don't know if you.
No, we are just starting with that. We don't have any results on that. We haven't tested any data on humans. But I think that it can, of course, AI will have, hopefully AI will have a big impact on this. Also, with all of these new studies on single cell, it might also help us understand better what's happening with interactions of the drugs with the cells and the resistance mechanisms and all of that, to see how we can overcome them, or even design new drugs that surpass those mechanisms and are able to attack those cells.
I think that I'm sorry. I think that I just remember one idea that it was in the way that when we talk about artificial intelligence and deep learning, it seems a lot like an abstraction, but in the end, it's just math and algorithms and codes. Okay, so and some of them they are more harder to understand than others, but it is not magic. It's math. But one interesting thing about this is that what these methods are enabling, it is not just to identify patterns on the data by themselves, but also to try to be able to identify what are the important features and how these features relate with the patterns. Let me give you one example. For several years, the development of new algorithms for image analysis, it was mostly done by trying to manually identify specific parts of the image of the photo or whatever that would relate with a specific class. And then, especially with the type of neural networks, convolutional neural networks, they were able to automatically identify what would be those patterns in the images. And one work that one of my students presented in, I don't know, a couple of years ago, it was the use of convolutional neural networks to try to identify on the protein and on the drugs, to identify these parts of the sequence that would be more important for the protein-drug interaction. And what we have showed, it was that we could have as good results that would be as good as with the use of the standard models for feature extraction from the proteins that were based on SAR and other strategies. So I think that one of the major things that neural networks and artificial is enabled is also to automatize this feature extraction and the analysis of what is relevant. So actually, when we apply these methods to images, we are just looking to the pixels, to the specific pixels on the image. And when we are applying this to proteins and to the letters, or amino acid letters that compose the sequence. And I think that this is also one of the major contributions that we are seeing today. And so, answering to the question, I think that this will also be one of the key features to have this impact.
So we are.
Sorry if I'm too long.
I can just add that, as you saw in my presentation, those strategies we are learning is that there are many things in the genome that are contributing to something. And this is the identification of these features that can, in all fields, lead to the selection of the parameters that should be observed for each person individually, and then feed the algorithms with that and say, okay, for this person, this will be the cocktail, for that person, that will be the cocktail, and not just one.
Yeah, of chemotherapy. And thank you so much. We're coming to a closure. Let me just check. We have here Inés asking about the supervised models. Rita also asking for some suggestions for someone who wants to enter this research field. So I will just do a very final brief round with Oscar, Joel, and Anna, and thank you once more for being here today.
This is a place for bioengineering. Just one thing that you need to know is that data analysis is about statistics, machine learning is about statistics and coding, so you need to have these strong backgrounds. Even coming from a BIOS star field, I don't care, you need to learn how to program, even from technical, that you can, the books that you can use to start understanding. And then it's just a topic.
I completely agree. I think that having a strong background in statistics algorithms, and then moving to programming, then to machine learning. It is the rock from the work that you are, the foundations of the work that you are doing.
Yeah. So, and all of bioinformatics, mathematics is very important, of course. Not just learning Python. If you don't understand some basic algebraic functions, it's going to be harder for you to even to start coding. So doing some online courses, having a strong background on maths, it's important.
Thank you once more. I don't know if anyone wants to add anything.
I just want to ask, what projects are being developed in education?
Wow, so many.
No, but in your area, not all.
Yes.
Rui, you can start. You have now.
Yeah, so we have this partnership with VIII where we are looking to new immunotherapies for instance, colorectal cancer, and being really targeting the more aggressive cases based on abnormal patterns of glycosylation on proteins. We want also to move to the epigenomic layer, where we want to see the impact of specific markers related with histone modifications and also the binding of non-coding RNAs, and how that can be used in order to enhance our knowledge of cancer and help with therapies. Oh my God, there's so many. This project on neurobiology, where we are learning predictors between tissues in the body, so we can understand potential hidden biological links between regulatory activity in different parts of our body. We've been also working with SARS-CoV-2, the microbiome in animals, and just one last project that I found it very interesting. It was one with Champalimo and Tekever. It's more on neurofeedback that I think is very interesting, where we wanted people to learn how to pilot drones, UVs, just with their brains. So we have a EEG cap, and we would create certain brain patterns to simulate different comments. So the implications in terms of health, we would also work with people with locked-in syndrome to remotely control interfaces, just with the power of the mind. And of course, we know that sometimes we have certain mental states that we would like to counteract. So this area of neurofeedback is very important in order for us to untap the potential of our brain. So that project was very interesting because we have a game, and for instance, if we want to turn right the drone, we would have, for instance, let's produce certain waves in this part of the brain and another waves on that part of the brain. And then we had the ball, and when somebody was able to simulate that brain patterning, the ball would go up. Okay, and some of us would easily put the ball up than others, and sometimes we feel a little dizzy. So we could really see the impact of neuromodulating our brains, and that area is very, very, I feel very promising, having like this neurofeedback, understanding what's going on in our brain, and producing new patterns that's being used for TDAH by stimulating the frontal lobe. Yeah, so those are just a sneak.
I can tell you that now at INESC-ID we have a thematic line that I'm coordinating, that is life and health technologies.
Yes, I went to a visit, and Luna also explained that. And
so we will start a large project on One Health concept that will be a partnership with the Faculdade of Medicina de Lisboa. Okay, One Health means that we want to treat patients with the most common comorbidities like hypertension, diabetes, but looking to the patient and seeing what that person is eating, where she's living, which are the risk factors she has, So this is a really huge project because we will have an hospital in Torres Vedras dedicated to the One Health concept. So this is something that we will push forward. That is our magical hospital. So we're starting is at the beginning, but now we are submitting projects on microbiome analysis, polygenic risk. So I think I saw my student over there. I'm not paying too much attention to Mathilde now because she knows that I'm submitting a project in the beginning of April. It is a huge consortium to integrate healthcare data in Alzheimer, cardiovascular, rheumatoid arthritis, and by integrating the data, improving the tools for medical doctors on diagnosis and prognosis. So
I'm at computer and electrical engineering, so I'm not from biology engineering. How could an electrical engineer contribute to this project?
to the One Health project in particular, we have in the Life and Health Technologies thematic line people from that are doing the electronics, for example, devices to monitor the patients, or even some devices to monitor. That is something that we will consider, or to use interactive tools for the patients to contribute with their own data when they're waiting for a consultation. Okay, but if you are in electronics, I'm an electronic engineer, my background. So it means that you can do anything you want in life. Sorry?
I'm still on my bachelor, but I was thinking doing my master in electronics.
Okay. But your bachelor is in what?
Electrical and computer engineering.
Okay.
You are welcome.
But you want
to know our
to
labs.
electronics.
Yes, in master.
Okay.
Yeah, we have
So
also
then we have those. We will develop tracking devices and using biosensors.
biosensors. So we are a little way
Yeah.
above time.
Yes.
So thank you once more for being here this morning and for our panel and for the wonderful organization.
Just one little thing before the session, I just wanted to say that the winner of the giveaway is Miguel Nunes. Is he here? So